Good morning, Doc. Good morning, everyone. So, together with my partner, Ma'am Daryl Grace Perwello, we are assigned to report to you about translation, gene expression. So, translation is the process by which the genetic code contained within a mRNA molecule or messenger RNA is decoded to produce a specific sequence of amino acids in a polypeptide chain. Translation is the language of nucleic acids translated into the language of proteins. So here we have four players. So these players are the key components which is required for translation to happen. So these are the mRNA or the messenger RNA, the ribosomes, the tRNA or the transfer RNA, and the amino acids. Okay, so first is the mRNA. mRNA is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene. So here, mRNA is the starting point of translation and is a temporary copy of part of a DNA strand. And also, mRNA acts as an instruction for the cell to build polypeptides. Then, in a mRNA, the instructions for building a polypeptide are RNA nucleotides, so which is read in groups of three. So these groups of three are called codons. So each codon corresponds to one amino acid, and the full set of codons is called the genetic code. Next, so the collection of codon amino acid relationships is called the genetic code. So because it lets cells decode an mRNA into a chain of amino acids. So the role of mRNA in translation is to tell the ribosome what amino acids are needed in a specific protein and what order to put them in. Okay, we have next here is the ribosome. So ribosomes structures where polypeptides or proteins are built and is made up of protein and RNA. So our ribosomal RNA or rRNA. So here in a ribosome, translation takes place inside its structure. Then, ribosomes' function is to manufacture proteins. So, ribosomes also translate information encoded in mRNA to assemble amino acids into a polypeptide chain. So, this complex is a macromolecular machine that can work together with the RNAs to convert the mRNA into polypeptides. So that the RNA and ribosomes work together to translate a sequence of mRNA into the corresponding polypeptide. So it carries a specific amino acid to the ribosome to help build proteins. Okay, next we have here is the tRNA or the transfer RNA. So tRNA is the molecular bridges that connect mRNA codons to the amino acids they encode. So the job of tRNA is to read message of nucleic acids or nucleotides and translate it into proteins or amino acids. So one end of each tRNA has a sequence of three nucleotides called an anticodon, which can bind to specific mRNA codons. Then the other end of the tRNA carries the amino acid specified by the codons. Okay, and lastly, we have here the amino acids. So there are 20 amino acids, each with basic structure. Amino acids are held together by peptide bonds. So we all know that amino acids are the building blocks of all biological proteins. So in translation, the amino acid regulate the translation of mRNA, and act to cause of preferential changes in the translation of mRNAs encoding particular proteins or families of proteins. So that are the four players in the translation process, which is the mRNA, the ribosome, the RNA, and then the amino acids. So here we have translation has three steps. So first is the initiation, second elongation, and the third is the termination. So this will be discussed to you by my partner, Ma'am Darl Grace S. Paruelo.
Thank you, Ma'am Diane. Good morning, Doc. Good morning, teachers. So my task is to discuss the three steps of translation. So the first one is initiation, or simply what we call the starting off. So during this stage, the small subunit of the ribosome first binds to the mRNA or the messenger RNA and moves along until it finds the start codon. Then, this codon is the sequence AUG, which corresponds to the amino acid methionine. So once bound, the transfer RNA or tRNA carrying methionine associates with the small subunit of the ribosome. So the next step is elongation, or simply what we call the extending of the chain. So during this stage, where the amino acid chain gets longer, in elongation, the mRNA is read one codon at a time, and the amino acid matching each codon is added to a growing protein chain. So once the tRNA carrying methionine binds to the start codon, the large subunit joins the small subunit, and the elongation stage of translation can begin. So the large subunit contains the three sites that the tRNA can interact with. The A site is the amino acid site, and the P site is the peptidyl site, and the E site is the exit site. When the large subunit joins, the tRNA bound to the start codon will be found in the P site. The next tRNA carrying the appropriate amino acid then associates with the mRNA in the amino acid site. Now, the ribosome can bring together the two amino acids to form a peptide bond with its peptidyl transferase enzymatic activity located in the large ribosomal subunit. This can link the next amino acid with the previous amino acid to elongate the polypeptide. Take note that a peptidyl transferase is the enzymatic activity of the ribosome, and it catalyzes the formation of a peptide bond by linking amino acids to the growing polypeptide during translation. Next, the ribosome moves in the 5' to 3' direction down to the mRNA to the next codon. This shifts the tRNAs into new sites in the ribosome. The tRNA that had lost its amino acid in the previous step now enters the E site and exits the ribosome. The tRNA with the polypeptide chain moves into the peptidyl site, and the new tRNA that contains the amino acid for the next codon enters at the amino acid site. To summarize the stage of elongation, first, the anticodon of an incoming tRNA pairs with the mRNA codon exposed in the A site. Next, a peptide bond is formed between the new amino acid in the A site and the previously added amino acid in the P site, transferring the polypeptide from the P site to the A site. Then, the ribosome moves one codon down to the mRNA. The tRNA in the A site carrying the polypeptide shifts to the P site. The tRNA in the P site shifts to the E site and exits the ribosome. So during the elongation, tRNAs move through the A, P, and E sites of the ribosome, as shown on this figure. So this process repeats many times as new codons are read and new amino acids are added to the chain. And the last stage is termination, the finishing stage of translation. So in the stage of translation, in which the finished polypeptide chain is released, these steps repeat and the polypeptide chain continues to elongate until a stop codon UAA, UAG, or UGA is released. So the translation termination is triggered by special proteins called release factors that cause the ribosome to release the polypeptide and the mRNA. The polypeptide can then go on to fold into its appropriate shape. After termination, the polypeptide may still need to fold into the right 3D shape, undergo processing such as the removal of amino acids, get shipped to the right place in the cell, or combine with other polypeptides before it can do its job as a functional protein. So once stop codon is reached, elements disassemble. So we have here a genetic code table. Each three-letter sequence of mRNA nucleotides corresponds to a specific amino acid, or to a stop codon we have UGA, UAA, and UAG are stop codons, while AUG is the codon for methionine and is also the start codon. So there are 61 codons for amino acids, and each of them is read to specify a certain amino acid out of the 20 commonly found in proteins. So one codon, AUG, specifies the amino acid methionine and also acts as a start codon to signal the start of protein construction, while there are three more codons that do not specify amino acids. These stop codons are UAA, UAG, and UGA. Tell the cell when a polypeptide is complete. So altogether, this collection of codon amino acid relationships is called the genetic code because it lets cell decode an mRNA into a chain of amino acids. Now, where does translation occur? So, for eukaryotic cell, the mRNA is transcribed in the nucleus and then exits through the nuclear pores into the cytoplasm, where the translation occurs. And for the prokaryotic cell, translation takes place in the cytoplasm. So both eukaryotic cell and prokaryotic cell the translation occur at cytoplasm. So for conclusion, translation is the process of converting a sequence of mRNA into a polypeptide. The site of protein is the ribosome in the cytoplasm. The transfer RNA is able to bring amino acids to the ribosome for translation, and translation goes through several stages in order to elongate a polypeptide. The first stage is the initiation stage, involves the binding of ribosome to the mRNA. Second is the elongation stage, involves extending the polypeptide chain. And the last stage is the termination. It occurs once a stop codon reach and the ribosome and polypeptide dissociate with the aid of release factors. Okay, so that is all about translation. Thank you for listening.